All right, students, as you know, AP Biology is a class about science, and science relies on experimental evidence in order to give us the information that we all now know and love. But those experiments, not only are they assessed a lot by the exam, but have certain things to keep in mind in order to make sure you're doing a good one, and how to interpret others is another great skill that we're going to work on today. So we're going to talk about experiment stuff. First and foremost, all good experiments have a control. And what I mean by a control is usually a group that gets everything else constant except for something to either determine whether it is, we're actually measuring the effect of the independent variable, or that the experiment works at all. And the positive control versus the negative control are going to be how you get there. So positive is that experimental group where you get a positive result at the end of the experiment. So you make sure that it's actually working. If you know the result, you're going to give a drug that you know the outcome of. That could be your positive control, and it ensures the success of the test. A negative control is a control that does not give a response to the test. That way you can show that when you give your independent variable, whatever you're directly changing, if you see a response, you go, okay, good, this is actually due to the independent variable. So it's like a saline shot or a placebo. You would have a group that does not give the experimental treatment. They get something that's very similar, but doesn't actually have the chemicals involved necessarily, just to make sure that the effect you see in the dependent variable is only reliant on the independent variable. Something else is when you are done and you have your findings, you need to report your findings. And those findings can be reported on a graph, some sort of figure. That's oftentimes how you're going to do it. When you do a graph, you're always going to want to put what you have changed directly or allowed to change during the experiment in a known way, such as time, as the independent variable here on the x-axis. And then the dependent variable, you're going to want to put here on the y-axis. Basically, what you're measuring, what you are looking at to see the effect on is going to be your dependent variable. It always depends on the independent variable, or you suspect that it's depends on the independent variable. If you see no differences between groups or across time, then it likely signals that it does not depend on it. But if you suspect it's going to depend on the independent variable, you're going to put it here on the y-axis, while your independent variable is going to be on the x. And then, and we're going to talk about this more, but if you have data and you're going to put it in a graph, you should have multiple trials. And when you have multiple trials, you should know if those different groups are statistically different from one another. And one way to tell that is with error bars. Remember, overlapping error bars mean that they are not statistically different. So we see 2 and 3 here are, their means on the bars are going to be the same. But as far as these two data sets, they are not statistically different from one another because they have overlapping error bars. This bar overlaps with this. This bar overlaps with this. Another example, it could be in, these are 3 chemicals in a paper that I pulled out of. You can see all of these groups are not statistically different because all of them have overlapping error bars. You can also see that with, it could be a line graph like this. It doesn't just have to be a bar graph that has error bars as well. As you can see at 30 degrees here, this is statistically different from these groups, the 20 degrees or the 24 degrees, for this experiment that went through time. And again, the dependent variable is going to be calcium sensitivity for here, and a mean VO2 here. And then the independent variable is going to be the main matrix or the time here. So those are graphs. Do I have any questions about graphs? Okay. Then we're going to get rocking and rolling on chi-squared. So with chi-squared tests, and we'll do a practice, you are trying to see if the group, the observed information, is statistically different from your expected. You can, we're going to do one where it's an ecology one, and we will just treat the expected as the mean, which is a fine thing to use. And that way, if you get a difference, you know that there is an actual difference between the groups. Whereas sometimes, and we'll do a brief one, if it's one with genetics, you've got homozygous or heterozygous information in the problem, then you're just going to use, instead of the mean, you just use the expected genotypic ratios. But again, it's exactly the same sort of overall process. It's just your expected might be a little bit different. In this case, it was a phenotype based on genotypes, and the experiment had an expected of 150 for red eyes and 50 for white eyes. So the calculation was done. There's two groups. So there was 1 degree of freedom. Don't forget that the degrees of freedom are the number of categories minus 1. And then you compare it to the table. This is where your degrees of freedom are. And we always use 0.05 for a p-value. Hey, hello. You must have just finished up. So we're just getting to talking about chi-square. So time works out pretty darn well. I just got a few in-person people here, video watchers that are coming on in to talk about chi-square. So again, what's that? Nope. I know. So again, we are, and we'll do a sample problem where we do use it like ecology. This case, it was not used like ecology. It was used like genetics, and we're going to talk about that. But overall, you do the calculation, you figure out what your expected is, you compare it to your observed, you set up the table, and you do the calculation. And remember that if the chi-squared value is greater than your critical value, which is your table value, then you're going to reject the null hypothesis. So this experiment, where it was red eyes compared to white eyes, first of all, there are 2 degrees of freedom, again, because it's the number of categories minus 1. So we would look in our table. This is on the actual equation sheet. Here, we're going to compare to a p-value of 0.05, and we're going to compare it here. So if we calculated out our value, and we got a chi-squared value of 6.83 compared to 3.84, it is greater than the chi-squared value, or sorry, the critical value. So we would reject the null hypothesis, which is likely that they are not, or probably the null hypothesis would be that they are not different than their expected values. So we can reject it in this case. So let's do a practice sample problem. So we'll do this one. I will do it at the same time as you all, and then we'll talk through it. An ecologist is testing habitat preferences for periwinkles on the rocky coastline of New England coast. She hypothesizes that more periwinkles will be found closer to the tide line. To test her hypothesis, she collects data by counting the number of periwinkles within the sample area that she observes on a rocky coastline. So these are the numbers, and we are going to do this calculation. So I want you to try it first, and then we'll talk through it. Mhm. Yeah. I think it's either the green sheet or the red sheet right over there. And I'll pull it up here too. Sure. To find the critical value, oops, I wrote down the wrong number. You figure out your number of degrees of freedom, which are always the number of categories you have, -1. And then you look in the area that has a p-value of 0.05. Yep, you're given this one here. So this is what it will look like on the actual equation sheet. Let me know when you're finished up. Okay. Are you good? Here, I'll pause it. Zoom recording. Okay, so we're gonna go over it. Let me line this up. Okay, so these were the observed numbers. Yes. Oh, funny. I'm very sorry. Thought it was down. I apologize. That might have been up during multiple classes today and nobody mentioned it. So. All right. So these were the observed numbers, right? At tide line, each of these meters above the tide line. And then big part of getting these questions right is figuring out what the expected is. I'm going to give you a problem where the expected is not this way. But if you don't know what the expected is, you should find the overall mean of whatever you're looking at relative to the total. So in this case, it was 75 total observed over the 5 locations. So 75 divided by 5 is 15, which is what we used for our expected values. Then we subtract our observed minus our expected. We get 21, 9, negative 5, negative 12, negative 13. And you may be worried about these negative numbers, but luckily enough, this is built right into the equation, is to square them, which is what we did here. So 21 squared is 441, negative 5 squared is 25, so on and so forth. And the last thing you do is divide by the expected. You normalize to your expected size. So 441 divided by 15 is 96.07. 81 divided by 15 is 5.4. At least according to my calculations. Do you have a different number? What did you get? Could be. Divided by 15. I did get a 29.4. Okay, good catch. So 29.4. I don't think it's going to majorly change my I mean, it'll change my answer, but I don't think it'll change the outcome. So it is 29.4. Let me check the other ones just to make sure that I didn't goof. 5.4, that one seems right. 144 divided by 15, 9.6, and 169 divided by 15 is this. Yep, so that is going to change that. And then that is also going to change this. So I'll add them back together, 29.4 plus 5.4 plus 1.67 plus 9.6 plus 11.27 gives me a chi-squared value of 57.34. So it definitely changes my chi-squared value. Be very careful about your calculations that you've made, but I think it still holds true. Then what you need to figure out is your critical value. And to do that, you're going to use this table. Degrees of freedom are always equal to the number of categories minus 1. So because we have 5 categories, 1, 2, 3, 4, 5, then our degrees of freedom is 4. And then we're going to compare it to this value because we're looking in this row. A 95% confidence is totally fine, and we get a value of 9.49. So we can see, I'll exit out of here, come back in here, that our chi-squared value of 57.34 is greater than the critical value of 9.49. So because of that, we can reject the null hypothesis. The null hypothesis would have been, or sometimes called the H naught, would have been, there is no statistical difference between the location and locations in terms of periwinkle numbers. Great name, periwinkles, by the way. Or you could say, following this conclusion, reject the null hypothesis. There is a statistically significant difference between periwinkle numbers and location relative to the tide line. Okay, does that make sense? Okay, just probably needs a little bit of brushing up on. And then I'm going to do one more practice where I'm basically just going to go over it. So these can be a little bit tricky, and you might get one about genetics and chi-square, but really there's not much difference. So for this problem, I'm going to go through this problem. And it's the same. I'll walk you through the parts that are maybe a little bit foreign, and then you can do the rest. So in this, multiple couples live in a small village on the eastern African lowlands, all of which are heterozygous for the HBS allele. So heterozygous, we'll call it just capital H, lowercase h for heterozygous. And I'll write, after we read through the problem, I'll show you what I'm writing down. HH. Of these children, they have 500 children among them. Of these children, 139 are homozygous, 279 are heterozygous, and 82 suffer from the actual disease. Are these statistically significant data is what they're asking. So let me show you how I would set this up. So 500 total. We have 139 that are, I'll write it like this. HH 139, H little h, we have 279, and little h are 82. Okay? That was observed. Okay, let me show you my calculations. Okay, so we actually observed out of the total, these were the numbers that were observed for each of these genotypes. And we need to figure out what the expected is. And this is the only tricky part of doing these relative to the other type. And the only thing is, if it's genetics, you should be thinking Punnett Square. Okay, so we have big H, little h, because all parents are heterozygous. That means we're going to get homozygous dominant, homozygous recessive, or sorry, heterozygous, and homozygous recessive here, which is for big H, we're going to have 1/4 of them. For heterozygous, we should expect half of them. And for homozygous recessive, we should expect 1/4 of them. Okay, and that's 1/4 of 500, which is going to equal, I'll just write it, 1 divided by 4 times 500, which is 125. Half of 500 is 250. And then homozygous recessive is again 125. Okay, so that's the only difference, is you're going to use a periodic table and then multiply the ratios times the total. That gives you expected. And then I'm going to have you guys finish out the rest of the problem. compare your answers. And I'm gonna stop showing my screen briefly while you guys do that practice. FRQ today is a relatively short one, so. Yeah, all right. Okay, so I got my chi-squared value. Hopefully you've got your chi-squared value. I got 19.724 with these expected values. Does anybody want to check my math to see if is that what you guys? Okay, cool. So then what value are we going to compare to on the periodic table, or on the periodic table, on the chi-square table? Why? Yep. Yeah. So it's we're going to compare to 5.99 because we have 3 categories minus 1 to get 2 degrees of freedom. We're going to compare to the p-value of 0.00 or 0.05, which gives us 5.99. So we compare 19.724 to 5.99, and we should see that this is greater. So what does that mean? Yep, you can reject the null again. Yeah. Yeah, just gotta shake off the reject the null hypothesis again. So there is a statistical difference. So something's going on here. Statistic. Why can't I remember statistical? There we are. Difference. Okay. So those are the big things that I want you to be able to do. And then I'm going to give you guys the FRQ. So I'm going to stop